Okay, so my topic is on baskets and umbrellas, and I'll be talking about clinical trials in glomerular diseases. My name is Syeda Ahmad, and I work at the Montlake Center. So I have no disclosures, and so we'll just do a little overview of clinical trials in nephrology, focusing more on glomerular diseases, talk about some barriers in nephrology, role of registries, industry partners, surrogate endpoints, in particular proteinuria, and what the title alludes to, master protocols, which are the future, which may be the future, including baskets and umbrellas. So this is just for definition purposes. Randomized controlled trials are typically classically identified as prospective studies to study the effectiveness of a new intervention or treatment. And we typically think of control versus treatment arm. So your control can be your placebo, which is a sham treatment, and we're looking to see if our experimental drug is superior to the placebo. But it can be inappropriate to use a placebo when there's already a treatment on the market that improves an outcome that is available. Then there's also another type of control, which is your standard of care. And this is when we look at a superiority study to see if our standard of care, if the study drug is superior to our standard of care, or whether it's an equivalent study, which is like a non-superiority study. And so historically, there was an article in JASN that was published in 2004 that looked at clinical trials in nephrology. And it said that nephrology was behind other specialties in randomized clinical trials. And what they did in this study was they did a Cochrane renal group specialized register and looked at randomized clinical trials, and they selected randomly 40 clinical trials for every 10-year period of 116 total. And they extracted data on quality of reporting by authors in dialysis, transplant, general nephrology. And they also compared articles that have been published in 2003 to 2004 in the major nephrology journals, which were JASN, Kidney International, and I believe HAKD as well. And the authors manually went through those articles that were published to see how many were clinical trials. To keep in mind before I present the results of this study, in 1962, the FDA had passed the drug efficacy amendments, and it wasn't until 2011 that rituximab was approved for ANCA vasculitis, which is specific to glomerular diseases, but it's specific to glomerulonephritis. It was more of a general general autoimmune disease. And then in 2020, Benlysta was approved for lupus nephritis, and in 2021, Tarpeyo for IgA. So there's a huge gap from when the drug efficacy amendments were passed until drugs were actually approved by the FDA, in particular glomerular disease. And so when you look at the data that the authors in that 2004 study looked at, they looked at, there were about 2,779 randomized controlled trials published in nephrology from 1966 to 2002, compared to 27,000 published in cardiology. And even though you notice an ascending trend in nephrology, it has been, this is a direct quote from this article, it has been slower than in other specialties. And this is where we are in nephrology as of 2004 when this article was published. And then they looked at the citations in the journals, and percentage of randomized controlled trials versus total citations in nephrology compared with 12 other specialties in internal medicine from 1966 to 2002. And they noticed that only 2,779 or 1.15% in nephrology were randomized controlled trials. And the discussion of this article has a pretty scathing analysis of nephrology. And so here we are, nephrology, not at the top. And so then they also looked at number of citations and number of randomized controlled trials in various areas within nephrology, and in the 10 major areas of nephrology, and the slope of the line corresponds to the average proportion of citations that are randomized controlled trials across all areas of nephrology. And here we are in glomerular diseases, which is my area of subspecialty, and we're pretty low at the bottom there amongst nephrology itself. And so the authors said that our data on the quantity and quality of trials in nephrology is a major concern and suggests that clinical research in nephrology is in crisis, which is a pretty scathing analysis. So, when you look at this, you're like, well, why are there few trials at that time, in particular glomerular disease? And we'll talk about some barriers which were highlighted. So first of all, you have to look at limitations of randomized controlled trials. We have rare disease, which is glomerular disease, and you need to study multiple sites to enroll significant number of patients. You have to look at your inclusion and exclusion criteria. If it's too strict, can make it difficult to enroll eligible patients. And you have to balance that with the criteria needed to have the intervention's efficacy. So these would be our statistical analysis. Then you also have to look at your patients. These are people with rare disease, and they may hope that they have a clinical trial to get a novel therapy, but they can be randomized into a placebo arm. So if you're a 30-year-old who is diagnosed with IgA nephropathy and you're offered a clinical trial, there's a chance that you could just be getting placebo with, optimized RAS inhibition. And then you also have to define your endpoint. So what is it that we're measuring objectively, and can this be standardized across all patients in the trial? And, what is that endpoint? Is it our change in GFR? Is it proteinuria? Is it both? Do we look at biopsy findings? Are there other markers that we could use, in particular biomarkers? And so this has, been a challenge a little bit and continues to be somewhat in nephrology. And so some of the common pitfalls that are also barriers, this is across all institutions and clinical trials, is slow IRB approval for clinical trials. There's a lot of contract and subcontract ratifications, and it can take months to get a clinical trial started. And there can be recruitment shortfalls because there's an overestimation of available participants. And, there's also training that's needed for a PI or sub-I to do for clinical trial design and implementation. And so this article published in 2016 in CJASN kind of gave this roadmap of what is are issues in clinical trials in nephrology. So on one hand, you have your industry with potential novel compounds. You have academia looking at novel pathways. And then you have what they noted as one barrier was nephrology culture, where sometimes nephrologists are a little reluctant to let go of their patients and enroll in clinical trials, even if it's for a short amount of time. This has been mentioned in several articles, not just this one, about the culture in nephrology. And then there's also a lack of druggable targets. So when we're looking at endpoints, when we're looking at, are we looking at uh, uh, biomarkers? Are we looking at biopsy endpoints? So that has been also a challenge as well. And then, they also highlighted the need for possible solutions such as registries and looking at other surrogate endpoints. So when we look at funding, which is how, trials happen, the NIDDK and NIH fund majority of the glomerular disease research in the United States. The FDA and the American Society of Nephrology have established the Kidney Health Initiative, and that is the website for that if you're interested in looking into that more, which is which is a collaboration to help bring out points of areas of deficit that can be addressed in nephrology research. And then there's also NIH-funded consortia such as Neptune, which is the Nephrotic Syndrome Study Network, CureGN, and then there's non-glomerular disease consortiums and registries such as the Chronic Renal Insufficiency Cohort and chronic kidney disease in children study, and the human hereditary and healthy and health in Africa kidney disease research network. And so the issue that happens is that with NIH-funded consortia, the conception to funding of a consortium can take 2 to 2.5 years, and it's dependent on available or projected funds. If you look at an R1 grant, and a lot of those the time frame is much shorter. So there is a major lag time in getting some of these things started to get the framework going for glomerular disease trials. And then when we look at registries, registries are helpful in then determining disease prevalence. You can study the size of a population. You can look at disease characteristics, and you can see how biomarkers perform over time. You can also use a registry to develop, bring out therapeutic endpoints and evaluating human therapeutic targets. And it can be a way to access patient recruitment and retention of patients. And it can also be helpful for global treatment modalities and obtaining critical mass for sufficient power. So there are several glomerular disease registries, such as the Toronto Glomerulonephritis Registry, the British Columbia Glomerulonephritis Network and Registry, and the International Nephrotic Syndrome Network. The issues with registries is that sometimes there can be redundancy, so a patient can be enrolled in multiple registries. They are expensive. They do require a lot of manpower. They do require manpower and resources to be able to maintain and constantly update. And you have to be able to integrate some of these with your EMR, so it can be easily tracked. So then we look at, we talked about in the earlier pathway about industry. So industry partners can be helpful in facilitating proof of concept human studies. And you can, have some opportunity from the industry in glomerular disease in particular. But with the issue with clinical trials is that sometimes the budgets can fail to adequately recognize the effort needed to identify patients who will meet eligibility criteria for a trial. Clinical trials are a time commitment for the PI involved and also the other staff. The patients have to sometimes come in 3 or 4 hours for a monthly site visit. Are they going to be reimbursed for their travel time, their day off from work, and all of those things? So those all have to be kept in mind. And then, trial designs are often, one of the benefits of industry partner can be that trial designs can be improved with early input from basic and clinical scientists and clinicians, as well as patients. So this is your classic clinical trial. The alternate model in partnering with industry would be that an academic investigator serves as a sponsor of a study that is funded by industry. So this would be like a much smaller study of looking at a particular molecule with an industry partner in a small group of patients. But then you also run into the issue of intellectual property ownership in academia versus industry. And this can also sometimes bog things down. But in industry partners, we have in nephrology, we have the NephCure Kidney International. And this is a partnership to help reduce the drug development time from 15 years to 5 years. And so the other thing we want to talk about is what are our endpoints? So you do a clinical trial, and how do you define where you stop and what are you going to look at as your endpoint? So surrogate endpoints are used in therapeutic trials as a substitute for clinically meaningful endpoint, and they can enable a faster and cheaper drug development. And they're useful in treatment of early stages of a slowly progressive disease. And so classic example that we have in glomerular disease, you could consider IgA nephropathy, but you also need post-marketing trial additional data to verify the clinical benefit. And so this is where we see the changes in the eGFR slope in all of the drugs that have been approved for IgA nephropathy. A biomarker is slightly different than a surrogate endpoint. This is an indicator of a normal biologic process, and a biomarker cannot always serve as a surrogate endpoint because sometimes there can be interactions or effects from the drug itself that are not essentially predicted, where a biomarker may not be the ideal case for a surrogate endpoint. So this is kind of the case where we look at, for example, in IgA, you could think of like the anti-gliadin IgA1 levels that, some of the newer papers are publishing. That could be considered a biomarker, but is it a valid surrogate endpoint is a different question. So when we look at proteinuria, we're going to talk about IgA in particular because I think that's the classic one or easiest one we can talk about. And so, we talked about serum biomarkers a little bit already. And then, you also look at IgA, may change, the biopsy findings may change with therapy. So you can have early, inflammatory disease that you treat with prednisone and you rebiopsy a patient and it's maybe a little bit of chronic disease. So can we use a biopsy finding as a reason or a surrogate as an endpoint? It remains to be seen. But then when you look at proteinuria, there has been evidence to show that urinary proteins from patients with nephrotic syndrome induce inflammatory and cytotoxic process in vitro, and this is thought to contribute to interstitial fibrosis, and that kidney tubular interstitial tissue from patients with IgA can be distinguished from control tissue on the basis of in vitro derived albumin-regulated genes. And so when we look at registries have been helpful in this. This is the data from the Toronto registry looking at time average proteinuria in patients with IgA nephropathy. And this was published in 2007. And this is clearly showing that patients with greater amount of proteinuria have poorer survival from renal failure. And then the RADAR registry, this was published in CJSN, I believe it's maybe 3 or 4 years now, showed that in patients, 2,300 adults with biopsy-proven IgA, even proteinuria greater than 1.76 at a GFR decline were going to get kidney failure within their lifetime. So, this has been important in IgA nephropathy. And so when we look at, how do you utilize all this data, the FDA in 2012 amended, Congress passed the Food and Drug Administration Safety Innovations Act. And this was to have accelerated approval for drugs for serious conditions that fill an unmet medical need, and looking at whether the drug has an effect on a surrogate or intermediate clinical endpoint. And so when we have proteinuria and we know that there is a correlation between eGFR decline, you can use that in IgA nephropathy, and the result has been pretty good in that we've had 4 FDA-approved drugs for IgA nephropathy since I finished my glomerular disease fellowship, so in the past 4.5, 5 years, which has been pretty phenomenal. But then you run into the issue of the problem with future trials. So is it appropriate to use a placebo when treatment that improves outcome is available? And this is looking at the 2025 KDIGO guidelines, which say use Nefogard or budesonide in patients with IgA. And then are we looking at precision medicine trials to evaluate targeted therapies? This can create challenges in recruiting patients with rare genetic subtypes of a disease and need to answer more questions more efficiently and in less time is also an issue. So what if we looked at more than 1 or 2 treatments in more than 1 patient within the same overall trial structure? And this is where master protocols come into play. And this was published by the FDA, in 2017, where we look at 1 or more interventions in multiple diseases, and each targeting a particular biomarker defined population or disease subtype. And there's 3 distinct entities: the umbrella, the basket, and platform trials. And they can involve direct comparison of competing therapies, and they can be structured to evaluate different therapies relative to their controls. And they take advantage of existing infrastructure that is available, such as similarities in trials, but it requires a lot of pre-trial discussion among sponsors. So the first master protocol we'll talk about is the umbrella trial. And this is looking at multiple target therapies in a single disease. And this will allow for faster identification of promising candidates to move forward in later stages of development. So for example, you have a single disease, you screen for presence of targets, and then they're going to go into biomarker 1 positive targeted therapy, 2, and then 3. And so an umbrella trial currently in glomerular disease is a phase 2A clinical trial called the RESULT study. And this is looking at FSGS and minimal change. And their cohort into this different ones, which is frexalimab. There is I believe this is like a TNF inhibitor type of molecule. And then this is a tyrosine kinase inhibitor. And they have them in different arms. So their patients are randomized into 3-to-1 ratio between the drug and a placebo. And when they do their statistical analysis, all the placebos are pooled together. And then they have an open label extension and then a follow-up. So in this particular study, the result, the trial that I just mentioned, the 3 drug candidates were already being tested in multiple diseases in phase 2 and 3 studies. So for example, I think hidradenitis suppurativa was one of the diseases that they already had data on. And so they did not have to do safety and pharmacokinetic data for glomerular diseases because that already had been completed. And they already had a chemistry, manufacturing, and controls process. And the project team was able to bypass the phase 1 healthy volunteer studies and go into the phase 2A efficacy signal-seeking trial. So the next trial we'll talk about is the basket trial, which is a single treatment across multiple diseases that share a common biomarker. So for example, you could study several pathologically distinct glomerular diseases characterized by shared feature proteinuria. And so you have, your 3, you could do IgA, minimal change, and, some another disease such as C3, screen for presence of a target, and then target positive participants. And so basket trials in glomerular disease, we have the atrasentan in patients with proteinuria glomerular disease, or the affinity study, which is looking at atrasentan, which is an ATA receptor antagonist in IgA, FSGS Alports, and also diabetic kidney disease on top of RAS inhibition SGLT2. There was also the RENEW trial looking at factor D inhibitor across membranous and in C3G and IgA, but this study was terminated. And then and so looking at the last trial we'll talk about is the platform trial, which is probably the most complicated of the 3, but it's also the most innovative in my opinion. And so study looking at multiple target therapies in a single disease, and therapies are allowed to enter, leave the platform based on a decision algorithm. So for example, if we look at a disease, we do screen, there's a continuous screening. And so you have biomarker stratum A start. And so patients are placed on standard of care versus investigational drug. And you can either stop it early because success is met, or you can introduce a new drug. If investigational drug 1 is shown to be superior to standard of care, it becomes the new standard of care. You can have patients in biomarker B, which is investigational drug 3 versus standard of care. Investigational drug 3 was no good. All of these patients are put into standard of care B. Then you have biomarker negative stratum. And so this is investigational drug 4 versus standard of care in patients who are biomarker negative. And then, you can also introduce other biomarkers as they come along. So an example of this is in oncology, the I-SPY2 study, which is new treatments for biomarker identified substates for early stage breast cancer. And so these were various drugs are tested as neoadjuvant treatments. So this allows for determination of primary outcome where they have the pathologic response of the tumor at surgery. And the patients are assigned to the most promising treatment based off of this or combination of treatments. So overall, they looked at 12 therapies from 9 sponsors, and 5 were advanced on for further study. And so master protocols have been used outside of oncology, such as in Alzheimer's, mental health, in a study of Ebola virus, and also in the antibiotic resistance leadership group. And so this has been very useful in non-oncology fields. It's an innovative way of using a trial network with infrastructure to streamline trial logistics, improve data quality, and facilitate data collection and sharing. You have a common protocol that involves innovative statistical approaches and enables a broader set of objectives to be met more effectively. And so, these are some of the areas of innovation where you have common screening, you have to have governance, and good infrastructure, trial networks and clinical centers, processes, randomization, quality control oversight, and then the trial design has to be adaptive with longitudinal modeling, and shared control patients, a natural history cohort, and biomarker qualification. So, things you have to consider, the time and resources to establish this infrastructure we're not there yet in nephrology. And increase upfront planning. So multiple parties need to be in agreement on trial design, operations, and governance. You need academics, for example, in I-SPY, they had the National Cancer Institute's pharmaceutical sponsors and the FDA. There has to be robust statistical models and algorithms and procedures to ensure rapid flow of information among the involved parties. And then, as the new drugs are approved, the standard of care can change, so it can impact a long-running master protocol. And one of the things with master protocols, and it's kind of in oncology, is some of these trials are just essentially never ending. But there was also an article in JAMA looking at the true economics of master protocols, and that's not really well established, whether to say that it's necessarily that much more cost effective than classical randomized controlled trials. So where are the future directions? We have newer therapies. So master trials are an innovative format to approach clinical trials in glomerular diseases. Since the article from 2004, we have made significant leaps in randomized controlled trials in nephrology. However, challenges still remain. One is funding. We have a changing nephrology workforce, with people, and so you also need education for trainees and junior faculty who are interested or may want to pursue this. And then support for investigators at academic institutions. So there is the International Society for Glomerular Disease. There is GlomCon, which has an annual meeting in Hawaii. If Raj is willing to sponsor me to go, it's week long. And then there is also ASN, NCAF, and NephCure. And then there's also the Glomerular Disease Center of Excellence establishment through the International Society of Glomerular Disease, which is looking at various factors. And, I think when I'm just looking at this figure, it's somewhat of I think in some academic institutions, even in like non-academic institutions, I think some of these things are going to be a little bit challenging because we are not at the same level as oncology and cardiology in the means of doing this scale of clinical trials. But this is, I think we're making a lot of progress, and I'll just open up to any questions that people may have.
Thank you so much, Syeda. Very, very good talk and very nice overview over about our clinical trial approach in glomerular disease. I think we do have a comment in the chat from Nisha. Nisha posted a recent paper discussing use of eGFR slope and decline in clinical trials, which I encourage folks to review. For the sake of time, I think we are just a little bit over. So I do want to give Brian ample time to present as well, but maybe we can continue any questions or discussion in the chat.